Good evening, ma'am.
Good evening. I will just share my screen and we will start when more people have joined.
Ma'am, please wait for 5 to 10 minutes.
Yeah, we will start in 5-10 minutes. Are you able to see my screen?
Yes, ma'am.
Okay, great.
So today we will solve the assignment, right? Because the answer key was already released.
Today we will be discussing questions from week 3. And from the assignment, if you have any doubts, we can discuss that. And there are some, I think, 3 reading assignments. Out of that, one paper also we are discussing in today's session. So today we have to be somewhat quick. We have to be little fast because this 10 questions we have to cover. And if there are any doubts from the previous assignment that we have to cover, and one more paper also we are covering today.
Ma'am? Hello, ma'am.
Hello, yes.
Yes, I am asking just about one thing. The exam mode in will be an MCQ only, right?
Yeah, most likely it will be MCQ only because all the previous year's questions and in all the NPTEL courses they are in MCQ only. So most likely this year also they will follow the same pattern. I think so. If there is any change, they will inform us. And if they give any information like that, I will inform in the class.
Okay.
Okay, let's get started now. I was just waiting for more people to join. Is my slides visible to everyone?
Yes, ma'am, visible.
Okay, great. So today, like the last session, we'll be discussing some MCQs based on the third week syllabus, and then we will discuss one reading assignment paper. So let's get started with the MCQs first. And if you have any doubts from the previous assignment, we can also discuss that after we discuss these questions. So, like the previous two classes, you can take 5 minutes to look at the question and think about it. And you can type the answer in the chat box, and then we will discuss the answer to the question.
I'm not able to see the screen.
Let me try resharing. Just give me a minute. How about now? Mm,
Yes, ma'am.
okay,
Ma'am,
great. Yeah.
is are the questions of the week 3?
Yes, this is from week 3.
Okay.
Okay, so the first question is what structures help in exerting pulling forces? So we'll go over all the options one by one. So option A, invadopodia, is like a structure with actin and some other proteins which can bind with actin and control their polymerization and rate of growth. And it is mostly found in the tumor cells, where tumor cells use these invadopodia to to basically go inside the extracellular matrix and go to migrate to other organs. And then there is philopodia. So philopodia are like very narrow and long actin stretches, and usually cells use these to probe the surroundings of the environment so that they can know that what is the stiffness in the surrounding area, etc. And based on that, they will choose which direction to migrate. And blebs are just whatever, like some small vesicle like structures found on the top of the cells. And option D, actomyosin contractility. So it is a combination of two proteins, actin and myosin. And this myosin is a protein which can bind to actin and it can pull on the actin fiber. So when this myosin pulls on the actin fiber, it will generate a force. And this force is what we call as actomyosin contractility. And cells use this mechanism to apply forces. I attached a video, but it's not playing. Yeah, it's here. So this maybe you can watch it later to understand how exactly this actin and myosin are interacting with each other to exert pulling forces. And the same mechanism is used in our muscles as well. So the answer to the first question is actin cytoskeleton.
Ma'am, the video we can access it later also, right?
Yeah, you can watch it later. Do you want to watch it now? We
No, no,
might
no, it's okay.
have time to discuss the paper, that's why.
Yes, ma'am, no problem.
Okay, cool. Let's move on to the second question then. So the second question is about RGD domain. So a domain is like a part of a protein, and many proteins, especially proteins which belong to similar families, they will be having these conserved domains which play some important function. So they are conserved within that family of proteins most likely. And this RGD domains, they are found in proteins of the extracellular matrix. Like for example, we have fibronectin, and they are also found in the proteins which interact with the ECM proteins. So, for example, they are found in integrin. So, RGD domain is mainly found in this context. So, RGD is just the sequence of amino acids, which is arginine, glycine, and aspartic acid. And so, they are found on both the ECM protein as well as with integrin, which binds to the ECM proteins. And the RGD domain, so mostly it will come in a combination, like one of, for example, the integrin, sorry, the ECM protein. If the ECM protein has the RGD domain, then the integrin will be having an RGD binding motif. So the RGD domain will bind to the RGD binding motif. So these will come in a pair. They two RGD domains can't bind with each other. They have to bind with their partner RGD binding domain. So, so these are, like I was saying, mainly present in the integrins and in the ECM protein. And integrins, you see there, like there are many kinds of integrins. So in integrins, they are a heterodimer, which have one alpha subdomain and one beta subdomain. And there are different classes of alpha and beta subdomains. And depending on which alpha and which beta subdomain come together, the integrin will be able to, some integrins can bind to collagen, some integrins can bind to laminin. That depends on the subdomains of the integrin. And some of these will be having the RGD domain. And sometimes the RGD domain will be found in the ECM protein as well. So here option 3 is not correct, because for collagen, so for fibronectin, laminin, and fibrinogen, for these ECM proteins, the integrin needs the RGD domain to bind. But for collagen, it has a separate collagen binding domain. So that's why option 3 is false. And the other options are all correct. Okay, let's move on to the third question.
Why is it not there for collagen?
So there is a different domain on the integrin proteins, not the RGD domain with which it binds with collagen. So it depends on the structure of the ECM proteins as well. So collagen has a very distinct structure from like fibronectin or fibrinogen. So the binding domain on integrin is also different. It has this triple helix structure, right?
Yes, ma'am.
Yeah. So the third question is, which of the following alpha integrins do not recognize collagen? So, like I was saying, so integrins are a huge family of proteins, and they are made up of an alpha and a beta subunit, and depending on which of these subunits come together, they can bind to certain ECM ligands, but not to others. And sorry. Yeah, so here, so the two main classes are beta 1 and beta 2 type. Like the alpha subunit is divided into two main classes, beta 1 and beta 2. And out of these, beta 1 class is what mainly binds, has collagen receptors and RGD receptors and laminin receptors. But alpha 5 has an RGD receptor, which so it is not able to bind to collagen. So the correct answer to this question is alpha 5, option B.
Like, how do we remember these alpha and beta and all that? Like this structure. How do we like remember it?
Uh, I mean, like, ideally, you wouldn't have to remember it because you can just look it up anytime. But I think from the point of view of the course, they might ask you questions. So yeah,
Maybe I think by answering questions we'll end up remembering it.
yeah, you could do that. Okay, let's go to the next question. So the fourth question is about the cell ECM interactions. How does the cell bind to the extracellular matrix? So the first point of contact is integrin, which has one part which is inside the cell and one part which is outside the cell, and the part that is outside the cell can bind to the ECM proteins, and the part that is inside the cell is connected to the cytoskeleton. So this connection between integrin and cytoskeleton is mediated by a series of proteins. It's not directly connected. So mainly, there are many proteins, but the main proteins are talin, vinculin, and then vinculin is then connected to the actomyosin skeleton. One special, and the reason that it's not connected directly, but it has these intermediate proteins is so that the cell can respond to external forces. For example, talon is a mechanosensor. So whenever there is an external force that is acting on the cell, it can extend talon, like it can unfold talon basically. So something similar to a spring. And when talon is unfolded, some new binding sites which were earlier hidden within, they are exposed when talon gets unfolded because of a force. And to these binding sites, vinculin can now come and attach. And then when vinculin comes and attaches, it increases again integrin binding to the ECM. So it's like a positive feedback loop, which helps the cells to react to external forces or to rigid extracellular matrix. And this is also the process by which focal adhesions are formed. We will come to focal adhesions in the upcoming questions. So the correct answer to this is option 2. Talon has exposed vinculin binding sites in response to external force. Yeah, so these were the proteins I was talking about. So first you have integrin, which is in contact with the extracellular matrix. And whenever there is an external force, the integrin goes from its folded to its elongated state. And when it goes to its elongated state, more proteins can come and bind to integrin, like talon and so yeah, talon firstly. And then talon also gets unfolded. And once talon is unfolded, vinculin can come and bind to talon. And then actomyosin fibers are formed at the vinculin junctions, and they exert a force on the substrate. So this is like the mechanism by which cells exert a higher traction force on stiffer substrates compared to soft substrates, which we were also discussing in the previous paper that we discussed last week. So the correct answer to this is option B. Talon has exposed vinculin binding sites in response to external force. Okay, let's go to the fifth question then. So the fifth question is, what is the resolution limit of an optical microscope? So microscopy is important in cell biology and in mechanobiology because uh, we use it widely to look at proteins, the dynamics of proteins inside cells. And in mechanobiology also, we use it extensively in like traction force microscopy and all. So this question is about the limitation of an optical microscope. So this limitation comes from the wavelength that we use actually. For an optical microscope, the minimum wavelength that we can go to is about 200 nanometers. So when these 2 points are at a distance of more than 200 nanometers, then the wavelengths won't get mixed up, and then we will see them as two separate dots. But when they are closer than 200 nanometer, because of the wavelength of light, the light waves coming from the first dot will interact with the light waves coming from the second dot. And instead of seeing them as two separate dots, we will see them as a single dot. So this is one limitation coming from the wavelength of the light microscope itself. So this is why, to avoid this, people have moved on to electron microscopy, because there the wavelength is so much lower and they are used to like visualize the structures of proteins as well. So the answer to this question is 200 nanometers. Let's go to the sixth question. So the sixth question is again about the focal adhesion. So these are the points of contact between the cell and the extracellular matrix. And using these points of contact, the cells are connected to the cytoskeleton, and they use it to exert forces on the extracellular matrix. So they are asking from outside to inside. So like we were discussing already, the outermost protein is integrin, and then the integrin is connected with a protein called focal adhesion kinase, which whenever integrin goes from its folded to unfolded form, FAK or focal adhesion kinase can bind to it. And after that, with focal adhesion kinase, paxillin is bound. And then with that, we have talin and vinculin, which we had discussed already. And after vinculin, we have actomyosin skeleton bundles, which are bound with vinculin. Yeah, and yeah, alpha actinin is, I can show you in the image. It's a protein uh, which can bind two actin fibers together. So because in focal adhesions, typically it's a large cluster of integrin molecules, and the actin fibers are also connected with each other to alpha actin. And then all of this, they become all these actin fibers, they come together and become a bigger bundle called the actin stress fibers. So the correct answer is option A. Let's go to the seventh question. So the seventh question is, inside focal adhesions, which layer is present on top of the integrin signaling layer? So this question can be taken in two ways, because the integrin signaling goes in two ways. It communicates, it goes from the ECM to the cells, which means that the cells can sense the substrate stiffness through integrin. And it also goes the other way. Cells can exert a force on the extracellular matrix using the integrin proteins. Um, so, um, so the first way, which means cell sensing the ECM stiffness, in that case, integrin is the like the first step. So there is nothing on top of integrin. So this question is about the second case where the cell is exerting a force on the extracellular matrix. So that originates from the actin contraction, actomyosin contractions. And from the actomyosin contractions, it goes to integrin, and then it becomes like the cell exerts a force on the extracellular matrix. So the correct answer is the actin regulatory layer. Let's go to the eighth question.
What do you mean by actin regulatory layer and post-transduction layer?
Um, so the actin regulatory layer is just the actin stress fibers which interacts with myosin to generate the contractile forces. And the force transduction layer is when the signal goes the other way from the ECM to the cell or to the nucleus actually, where the forces in at the extracellular matrix or outside of the cell is transduced into the mechanical forces are transduced into chemical signals using these focal adhesion proteins like vinculin, etc. So that is the force transduction layer.
Okay, thank you, ma'am.
Yeah. So this question is, which of these adhesion structures have the least number of focal adhesion proteins? So focal adhesions are not the only site of contact between the cell and the ECM. There are also other structures like focal complexes and fibrillar adhesions, which have different functions. So focal complexes are smaller than focal adhesions and they generate lesser force, and they are also less stable. So they only stay for a lesser time. And focal adhesions are the main sites where cells exert a force on the ECM as well as they can sense the ECM substrate. And fibrillar adhesions are where the cells are bound with the fibronectin substrate. And so here it doesn't have myosin; it only has actin. So fibrillar adhesion is not involved in force generation. It is just for the mechanical integrity of the cell. But fibrillar adhesions, they still have a lot of actin fibers which are bound together by another protein called tensin. So you get these stress fibers which are like the ropes of a tent, and to help hold the cell in place. But focal complexes, like I said, they are very small structures and they are very transient in nature. So they don't have a lot of proteins. They only have some proteins which are found in the focal adhesion. And which proteins exactly, that we'll discuss in the next question.
Ma'am, please move to the previous question. Please tell the reason why the focal complex is about the least number of focal adhesion proteins. Why not another things, focal adhesion or fibrillar adhesions?
So focal adhesion we were seeing here, right? It is a much larger structure. So these are about 2 to 10 micrometer square, and they have this like huge assembly of proteins. So it starts with one or two integrins, but whenever the cell is exerting a force or whenever the cell is on a stiffer ECM, it leads to the recruitment of more integrins and more of all of these adapter proteins like FAK, vinculin, talin. They all bind and they form this huge structure with which the cell can exert a force on the substrate. So this is the function of focal adhesion. So it is a large structure with a lot of proteins which can exert a force on the substrate. But focal complex are, yeah, it's here. Focal complexes are smaller, much smaller. So they are like 10 times smaller than focal adhesions, and they are also very transient. So focal complexes are like, there are many of them formed in the cell, but they are much smaller in number. Focal adhesions are opposite, like they are not as many as focal complexes, but they are bigger structures and they can exert a bigger force. So for focal complexes, the main function is not to exert force. It is just like, sorry, they can exert a force, but it's a smaller structures which exerts much lesser force than focal adhesions, and it has much lesser number of proteins. And fibrillar adhesions, they are more of a structural element where a lot of actin fibers are linked together with the help of this protein called tensin, and they form like stress fibers, something similar to a rope to hold the cell in place. So these are also still larger than focal complexes. Is it clear?
Uh, but the fibrillar and focal adhesions have more extensive protein, right? So, and in case of the focal complexes, the least smaller and mature adhesion structures. So that's the only reason.
Yes, yes.
Okay, now I can understand.
Okay, great. So like we were discussing, focal complexes are smaller and lesser proteins are recruited. So integrin is already there, like it is always there between the cell and the ECM. But when the cell has to exert some force, it can form focal complex or focal adhesion depending on how much force it has to exert and whether it needs to be there for a long time or not. So for focal complexes, mainly only paxillin and vinculin are recruited. If you look at the focal adhesion, then more proteins like talin, alpha actinin are also there in the focal adhesion. But in focal complex, it's mainly paxillin and vinculin that are recruited. So they're much smaller and they exert only very less forces and they are also less stable. Let's go to the 10th question. So this question is based on a concept that we have already discussed. So blood clotting means the platelets and other blood cells, they have to go and bind to the wall of the blood vessel so that there is no leakage of blood. And in the first question itself, we were first or second question, we were discussing that the RGD domain, with the help of the RGD domain, the cells can bind their integrin with the ECM protein. So that's why the answer to this is RGD domain. Yeah, the last three questions I just went a little bit fast because we have to discuss the papers as well. So I think there are these two papers and one more paper was mentioned in a different lecture, but we are just going to discuss the first paper today. This is the paper. You can take up to 7:30 to read the paper, and half an hour we can have a discussion here. This is the paper. I'll just put the link in the chat box. Are you able to access this link that I sent? You can see the paper from this link, and up to 7:30 you can read this paper and then we can discuss it. You have finished reading the paper? Hello?
Ma'am, please give me 10 minutes more.
Okay, fine.
Hello, ma'am. Hello.